I'll now introduce the last speaker of this session, Patrick Hay. Patrick is a postdoctoral researcher in the Respiratory Technology Group at Woodlock Institute of Medical Research. He received his PhD from the University of Technology in Sydney in 2023, and currently he's actively engaged in various projects involving drug delivery testing for inhaler devices and development of 3D respiratory models. So let's now hear from Patrick.
Thank you for the introduction, and thank you, DDO organizers, for inviting me to give this talk about assessing low-dose aerosolized toxins using the Advanced Integrated Respiratory Model, or AIR model for short. So first, I'd like to introduce the team that's been on this project, the members of the Woodlock Institute of Medical Research, our collaborators from Macquarie University, the University of New South Wales, and Australian Department of Defence. So we all here are constantly exposed to various toxins and toxicants from the air that we breathe, whether it is chemicals from cigarette smoke and burning fossil fuels, or harmful proteins that are produced by plants and bacteria. It is important for us to predict and evaluate injury to humans when inhaling these aerosolized toxins, to be able to understand where it goes in the body and also what it does to the body. To be able to do that, we require a 3D physiologically relevant model to be able to assess these parameters and also using these parameters to produce a strategy to predict and protect the public health. The current in vitro models, such as cascade impactors, are able to characterize aerosol physical properties, which means it looks at particle size distribution, also particle deposition. However, the biological effects of the air and aerosol with the cell interactions is unknown. Static models such as 2D and 3D cell culture models are able to understand the biologics, with 3D air-liquid interface models being more physiologically relevant in the cell physiology, where the cells are exposed to both the air and liquid. More advanced dynamic models, such as microfluidic chips, look into fluid flow, also mechanical stress in cell physiology. However, static and dynamic models are unable to look into what cascade impactors can, which is physical properties. Hence, we developed a 3D physiologically relevant model, which integrates both aerosol deposition profiling and also the biological effects. In this talk, I'll be comparing a 2D model versus the air model, and also using the air model to assess the transport of a model protein toxin, RCA1. So first, I want to look at the design of the air model. The air model design was based on two modeling techniques. The first one being an idolized technique. We adapted the lower respiratory tract from the Weibel's fourth generation model, generation 0 to generation 4, from the trachea down to the bronchioles. Then the upper respiratory tract was using the image-based technique. We used MRI scan of a healthy adult participant. And then the upper respiratory tract and lower respiratory tract was then reverse casted using silicon. Silicon was chosen for its inert and also transparent properties. The lower respiratory tract was then, as you can see, placed inside a plexiglass chamber simulating the lungs. Then we integrated in the airline models. So transwells can be placed in various regions of the air model, depending on what you want to test and which way you want to test. However, for this talk, I'm just going to be showing you data that where the transwells are placed in the terminal regions of the lower respiratory tract. So we want to characterize the air model. We use fluorescent sodium solution at 15 liters per minute using the PariJet nebulizer for 2 minutes. Because of its transparent properties, we can just visualize it initially. You can see the red arrows indicating where most of the deposition is occurring. From the top to bottom, we can see from the oral cavity, then the epiglottis, and then the terminal regions of the lower respiratory tract. Furthermore, we also saw a non-homogeneous distribution of the aerosols through the air model, which is a closer mimic of what would happen if you inhale these aerosols. Next, I'll show you a short clip showing the aerosols leaving the terminal regions. You can see there's the transwells attached onto the lower respiratory tract. We want to further characterize the air model, looking at the recovery and deposition using the Fluidnar solution. We can see at the air model itself, over 3 independent experiments, the recovery was at 100%, very consistent. And then we compared against 2 other parameters, the aerosols containing transwells and the transwells containing cells. We saw no significant difference. Next, we looked at the deposition profiles across the different regions of the air model. Just looking at the delivered dose, we see that the chamber showed highest deposition of lunar solution, followed by the mouthpiece throat and the lower airways. The presence of transwell membranes containing cells or without cells did not significantly alter the overall deposition profiles in the regions, showing that the experiment was consistent across the different parameters. Integration of the air-alive cellular model with the air model, we looked at H441 cells. H441 cells were used as a representative cell line for the distal region of the lungs. H441 cell lines were cultured on transwells for 14 days until confluency and then attached to the terminal regions of the lower respiratory tract, as indicated by the orange box. The RCA1 solution was then aerosolized into the air model as a model protein toxin for 15 liters per minute for 2 minutes. We looked at the paracellular permeability assay, so how permeable the fluoride solution was across the H441 monolayer post-treatment with RCA1. We then looked at the transepithelial electrical resistance, TER, which assesses the cell tight junctions and also the barrier function post-treatment with RCA1. And then finally, we looked at the rate of transport of RCA1 across the H441 monolayer, and an analysis was done by electrochemical. We want to understand and compare the toxic doses from a 2D model versus the air model. MTS assay was used as the 2D model. MTS assay being a cell viability assay, we looked at a range of RCA1 concentrations, which initially we got from literature. The IC50, or inhibitory concentration 50%, where 50% of the cell mitochondrial activity was inhibited, was deemed a toxic dose. Using the IC50, it was aerosolized also into the air model. As you can see, where it's outlined, we no longer saw a significant toxic dose. We can see that in the air model, compared to control, there was no significant difference. However, if you increase the IC50 by 10-fold, we see a significant drop in the electrical resistance, which is indicating that there is damage to the cell tight junctions and barrier function. Similarly, the results were shown for the permeability assay, where the IC50 showed no significant difference compared to control. However, when a 10-fold increase, we saw a significant increase in permeability. We next want to assess the rate of RCA1 transport in the H441 cell line. We looked at 2 doses, the non-toxic and toxic dose, which was previously attained from the TER and permeability results. At the non-toxic dose, we look at the 45-minute time mark, and we see it's roughly 25% of the RCA1 mass percentage has been delivered, or transported, sorry. And then when you compare it to the 2-hour time point, there's a significant increase. However, at the toxic dose, you see there's no significant increase anymore, with the 45-minute time mark around 50% of the RCA1 being transported compared to the 2-hour time point, which means there's a faster rate of transport at the toxic dose, which is indicating of that the permeability has been increased and also there's damage to the tight junctions. In summary, we characterize the air model and show that even with the transwells, with or without the cells, the recovery of fluoride solution was not significantly different and deposition was consistent. The 2D model compared to our air model showed vastly different toxic concentration thresholds, with the air model being a more discriminatory and physiologically relevant result, accurately depicting what the toxic dose and drug transport rates would be in human physiology. In this study, we just showed we tested toxins. However, the air model was able to assess a range of biological molecules, like for mRNA, for example. Thank you.
Thank you, Patrick, for your interesting presentation. Do we have any questions? All right, I might start just with a quick curiosity then. So in your model, you have evaluated the H441 cells. So are you planning to expand and to evaluate other kind of cell lines?
Yes, so H441s were initially looked at. We also looked at other cell lines like Cali3, which is more in the bronchial region, might be even more representative of the model that where the transwells are attached. We also gonna look at like other cells like Bs to Bs, A549s. Yeah.
Can I ask in your model of the transwells?
Yes.
You've got multiple transwells. So when you say n equals 3, is that various transwells in each n equals 1? Do you get multiple measurements each experiment? Is that how it works?
Yeah, so n equals 3 is independent, so that's only, so the transwells were just for one experiment, and then we averaged the data. And even though the transwells because of the non-homogeneous deposition, it's very different results between it. Yeah, so you can't put it as n equals 1 or else the results were pretty varied. Yeah, so that's only for one, n equals 1 is all of the collective transwells averaged.
And how varied is the dose delivered transwell to transwell within that one experiment?
So the dose, sorry.
The inhomogeneity, how different is the dose to each transwell within that n equals 1?
Right. So it's actually very different. So the inner transwells have a lot less compared to the outer transwells, almost 3 times less. Okay. And so because of how low the concentration it is, we have to use electrochemical analysis because HPLC can't even detect how low the doses of the inner transwells are.
Okay.
Oh, we have a question.
Thank you. How long can you keep the cells viable in the transwells in that system, and what are you doing to sort of feed them or keep them going? And related question, do you have any controls, like control groups to measure that?
Yeah, thanks. So yeah, we have control groups which we compare against, and how we keep it viable is because most of the tests are done, like the transfer is 2 hours, and every time we finish one test, what we do is put it back into an incubator, a non-sterile incubator, and then although it's non-sterile incubator, we know that bacteria won't start growing within the 2 hours. So that's why we have 2-hour transports and not really extended long transports.
We have time for a couple more questions. Right. So maybe,
Go ahead.
All right. So looking also at your deposition results, have you planned or have you evaluated to compare your results with some more conventional devices used to evaluate a deposition profile, like an NGI, let's say?
Yes. So we did look at NGI data. I didn't present NGI data today for RCA1 only because I haven't done the RCA1 for air model, so it would be not good if we don't compare both. But the RCA1 for NGI data is also hard to compare with the air model because we haven't fully characterized the actual particle size of the air model itself. So if an aerosol is deposited in the trachea, we don't know exactly what particle size, the cutoff diameter for that trachea region. We are actually currently just doing this experiment where we're going to add a sticky substance, like a sticky tape, and then we're going to see, once we aerosolize an aerosol and the deposits on the sticky substance, we're going to look at it under a microscope and evaluate how the diameter cutoffs that could be for the air region as well. So we can sort of understand what's the cutoff diameter and compare it to the NGI.
Thank you.
Thank you.
And in terms of the dosimetry, because you've got two very different systems you were comparing. You've got the 96-fold plate setup where you're exposing very controlled concentrations to all the different wells, versus when you've got your air model, you're depositing particles onto an air interfaced surface. So how do you compare sort of concentration? How do you sort of normalize the concentrations between those two very different systems?
Yeah, that's a really good question. So as you know, MTS is just directly added on, so but the concentration doesn't change, but the volume that deposits on the cells would change on the transwells. In our case, it's hard to normalize exactly how much we get from the transwells compared to the 2D model, but that's, I think, how important it is to understand when you're looking at the air model, the deposition process is so different. So MTS assay is just only a direct measurement of the concentration, but the air model you can see more than just direct measurement, but also how the physiological relevance it is.
All right, if there are not questions from the audience, I think we can thank Patrick again.